How exactly is food turned into energy? Well, the path is, as the Beatles sang, a long and winding road. And really, it's more than one. In this lecture, we'll travel the first biochemical leg of that journey. We'll spend most of our time driving a metabolic pathway called glycolysis that breaks up sugars, but we'll also loop around a smaller route called fatty acid oxidation. A metabolic pathway is a series of biochemical reactions where the product of one serves as the substrate for the next one. Each reaction is catalyzed by an enzyme, and each molecule is called an intermediate. Biochemists compare diagrams of metabolic pathways to roadmaps, and with good reason. Where cities are the connection points for highways on roadmaps, intermediates are connecting points for enzymes acting on them. A great feature of metabolic pathways is that they are essentially identical for humans, manta rays, or any organism we choose. Consequently, if you learn a metabolic pathway for one organism, you've already learned a pathway that's in almost every other organism. The advantage for understanding metabolism is huge, as you could imagine. While you might need a GPS to navigate the unique features of each highway, the only GPS you need for metabolic pathways is a general pathway scheme. Yep, that's right. There is a cellular GPS, and one size mostly fits all. The glycolysis pathway is like a journey along the I-5 freeway from Seattle to Los Angeles. Although it is clear that this can be seen as a single road, we know that other highways join I-5 along the way. So a person could get to LA without starting at Seattle. It's also possible to get off the freeway at various points and head someplace besides LA. Finally, the freeway continues beyond LA, passing through even more places with connections to additional big highways. Similarly, reactions can join the glycolysis pathway without starting from glucose and take several alternate connecting paths. I want you to see that when we talk of individual pathways with distinct starting and ending points, we're actually focusing on a single section of a bigger network. So while we are going to consider the main route of glycolysis here, it is important to remember glycolysis is only part of a bigger system. Cells act on glucose in 10 steps in glycolysis. Glucose contains 6 carbons and gets broken down to 2 identical molecules of pyruvate, each with 3 carbons. Other sugars can be oxidized in glycolysis as well. In the process, glucose gets oxidized. This involves release of energy, which is partly stored in the phosphate bonds of ATP. With this in mind, let's take a road trip through the 10 cities of the glycolysis pathway. The process all starts in the cytoplasm. In the first reaction, a phosphate from ATP is transferred onto carbon 6 of glucose to create a molecule known as glucose 6-phosphate, or G6P. This reaction requires ATP instead of producing it. Now, the expenditure of ATP energy to start glycolysis is sometimes compared to priming a pump. That is, to make ATP, we have to use some ATP. For cells, ATP is an investment strategy. It will pay dividends later. Enzymes that put phosphates onto molecules are kinases, and this enzyme is called hexokinase. Adding a phosphate onto glucose not only primes the pump, but it also makes the molecule negatively charged. Charged molecules cannot easily exit cells on their own, so the phosphate put onto glucose puts it under house arrest, so to speak. Step 2 in the process is a simple rearrangement of G6P. That is, there is nothing added or taken away. The atoms in the glucose 6-phosphate are simply rearranged to make fructose 6-phosphate, or F6P. This reaction is simple and unremarkable. F6P, though, is the substrate for the next reaction, reaction 3, a crucial control point for the pathway. In that step 3, a second ATP is invested to add a second phosphate onto F6P, this time at carbon number 1, making fructose 1,6-bisphosphate, or F16BP. The enzyme catalyzing the reaction is called phosphofructokinase, or as I'll more commonly say it, PFK. This enzyme is capable of controlling the entire pathway, as you will see. So 3 steps into the process, and we've done some rearranging of sugars, used up 2 ATPs to add a couple of phosphates. We're now at the end of the energy investment phase. Later in glycolysis, we'll recoup this energy investment and come out ahead, but we're still at 6 carbons. That changes in the next step, step 4. Catalyzed by the enzyme called aldolase, step 4 of glycolysis splits F16BP into 2 pieces of 3 carbons each. Now these 2 pieces are not identical, but they are instead isomers, meaning that one of them can be rearranged into the other. And that's actually what happens in step 5, which is the next step, where the piece called dihydroxyacetone phosphate, or DHAP, gets converted into its isomer glyceraldehyde 3-phosphate, or G3P. So basically after step 5, we have 2 molecules of G3P from the 1 starting molecule of glucose. Starting in step 6, glycolysis becomes a process of energy generation, where ATP is made. Now to begin this process, the glyceraldehyde 3-phosphates, or G3Ps, are converted to 1,3-bisphosphoglycerates. This involves the addition of another phosphate. For this to happen, the G3P aldehyde molecule is converted to an acid. Now when you convert something from an aldehyde to an acid, that's an oxidation. What else happens in an oxidation? That's right, the molecule being oxidized loses 2 electrons, and it gives those electrons to NAD+, which accepts them plus a proton and makes NADH. We had 2 glyceraldehyde 3-phosphate molecules, so we get 2 NADHs out of this reaction. 2 lectures from now, we'll see this energy used in electron transport to obtain a much bigger energy payoff. In the same reaction, a second thing happens. The acid group created by the oxidation reaction gets a phosphate linked to it, yielding 1,3-bisphosphoglycerate. The reaction is different from the phosphate additions that you've seen so far, however, that required ATP energy. This time the phosphate is not from ATP, but rather a free phosphate added onto the acid. Now the energy for doing this comes from the oxidation of the same step. And this is why we don't need ATP energy to add the phosphate. The net tally for this reaction is 2 1,3-bisphosphoglycerates and 2 NADHs. Now formation of 1,3-bisphosphoglycerate kicks off the last reactions of glycolysis, where we actually do begin to make ATP. First, a phosphate gets transferred away from each of the 1,3-bisphosphoglycerates to ADPs, so we make 2 ATPs. And we leave behind 2 molecules called 3-phosphoglycerate. So at step 7, we've broken even because we've gained 2 ATPs and recovered the energy invested in steps 1 and 3. In step 8, the molecules of 3-phosphoglycerate undergo rearrangement. And in step 9, they form a molecule called phosphoenol pyruvate, or PEP. PEP is the substrate for the 10th and final step of glycolysis that I call the Big Bang. The Big Bang reaction, which is catalyzed by the enzyme pyruvate kinase, gets its nickname from a very large energy release that results from the reaction. In the reaction, 2 phosphoenol pyruvates are converted to 2 pyruvates and 2 ATPs are produced. Now this reaction is energetic enough that there's enough energy released in the reaction to almost produce 2 more ATPs, but not quite. Energy released during reactions that is not captured or used, as in this reaction, are lost as heat. So though most metabolic reactions release some heat, this one produces more than most of them. That's one reason we get hot when we exercise. Cells are busy running heat-releasing reactions of metabolism. So we've broken down glucose into 2 pyruvates, but what have we achieved? When we do the tally, the products of glycolysis include 2 ATPs, after subtracting the 2 input ATPs, 2 NADHs, and 2 molecules of pyruvate. Now that may not look like a lot, but the payoff was double our investment. Moreover, the NADH made in glycolysis can be used to produce more ATPs, and the 2 molecules of pyruvate can be further oxidized in the citric acid cycle. In total, there is enough energy in one glucose molecule to give 32 to 38 ATPs. So glycolysis mostly just sets the stage for the main act of energy extraction, and we'll see how all that occurs over the next 2 lectures. Meanwhile, the 10 steps of glycolysis do more than get the breakdown of glucose started. Remember, every pathway is part of a bigger network. Intermediates of glycolysis are used in other metabolic pathways. So, for instance, G6P from step 1 can also enter the pentose phosphate pathway, which converts it into other sugars like ribose 5-phosphate, and that's needed to make DNA and RNA. Likewise, step 2's fructose 6-phosphate can be connected to proteins in the pathway of making glycoproteins to provide cellular ID tags. The PEP in step 9 is also used by some plants for extracting carbon dioxide from the atmosphere in photosynthesis. And pyruvate from step 10 is not only the gateway to the energy bonanza of the citric acid cycle, it's also a starting point for making the amino acid alanine. Yeast also use it to make ethanol. So in short, glycolysis not only doubles our energy investment, it also creates building blocks that can be tapped for making DNA, other sugars, amino acids, and more. Now, having multiple steps also provides several points at which the flow of intermediates through glycolysis can be adjusted. It's like a vacation where kids are better behaved if you stop along the way. Like traffic on busy streets, metabolic pathways must be regulated. So let's take a look at some of the stoplights along the way. Step 1 of the pathway, catalyzed as we saw by the enzyme hexokinase, makes glucose 6-phosphate from glucose. However, the enzyme is inhibited by high levels of the product, G6P. This feedback keeps cells from piling up large amounts of phosphorylated glucose. Step 3 of glycolysis was where the enzyme phosphofructokinase, or PFK, added a phosphate onto fructose 6-phosphate, making fructose 1,6-bisphosphate. It is the main stoplight for the pathway and is allosterically regulated by 3 different molecules. First, ATP. Second, a low-energy cousin called AMP. And third, a double phosphate molecule known as fructose 2,6-bisphosphate, or F26BP. As you learned from the lecture on enzyme regulation, allosteric regulating molecules bind to an enzyme and alter its activity. When any of these molecules binds to PFK, the resulting change in the enzyme's conformation dials its activity and traffic of the entire pathway up or down. An abundance of ATP indicates that the cell has plenty of energy. So when it binds to PFK, the enzyme's activity slows down. It's turned down. Large amounts of AMP, on the other hand, signal low energy and a need for glycolysis to make ATP, so things get turned up. Binding of AMP to PFK therefore increases PFK's activity. The other green light is F26BP. Now, F26BP sounds a lot like F16BP, which is made in glycolysis, but F26BP is not. It's different. F26BP is made by cells in response to insulin. Insulin stimulates cells to take up glucose following a meal. When this happens, they must run glycolysis to break it down. So F26BP increases PFK activity and speeds up glycolysis. In summary, PFK's enzyme activity has 2 accelerators and 1 brake to match the changing needs of cellular traffic. Regulation affects the aldolase reaction that splits glucose into DHAP and G3P, but not through control of its enzyme. This reaction would move backwards, not forwards, under standard conditions. But regulation of other enzymes in the pathway helps ensure conditions are not standard when glycolysis needs to run. Now, it does this by ensuring that the substrate, F16BP, is in high concentration, and the product's concentrations are kept low. Now, remember that cells must adjust molecules' concentrations to control reactions. It's easy to keep the levels of the substrate, F16BP, high as long as there's enough glucose flowing through glycolysis, and that happens when PFK is activated. So step 1, check. For products, cells decrease their concentrations by quickly converting them into something else. Now, this is not a simple one-step process in glycolysis, but it's a clever one relying on the connectedness of reactions in a pathway. It actually involves the substrate for the reaction, F16BP, and the final enzyme of the pathway, pyruvate kinase. Let's look at how that works. First, remember the products of the reaction, DHAP and G3P. In step 5, DHAP rapidly gets isomerized to G3P, so that latter molecule is all we really need to consider. G3P is used in step 6 to make 1,3-bisphosphoglycerate. Step 7 relies on step 6, step 8 relies on step 7, and this occurs all the way down to step 10. This chain gang of reactions moves toward whichever end of the chain gets pulled. We're going to focus on the end of that chain, the pyruvate kinase reaction. The final enzyme controlled in glycolysis is pyruvate kinase at step 10. Metabolic pathways do not typically have control mechanisms operating at the last reaction of the pathway, but in this case, it makes good cellular sense. The regulation of the pyruvate kinase depends on 2 mechanisms for feedback inhibition and 1 for activation. Both of the inhibition mechanisms involve byproducts of the pathway inhibiting the pyruvate kinase enzyme at the end of the pathway. The first inhibitor is ATP. If there's plenty of ATP, there isn't a need to break down sugar to make more. And where do we get ATP from? Glycolysis. So you may recall that PFK in step 3 was also inhibited by a surplus of ATP, so that's not a surprise. Pyruvate kinase is also inhibited by alanine. Alanine is an amino acid that is made from pyruvate, so a high concentration of alanine indicates an abundance of pyruvate. Turning off the pyruvate kinase makes sense under those conditions too. The inhibitors work as we might expect, but the one activator is more surprising. It's F16BP, the product of reaction 3 and the substrate for reaction 4. Why would pyruvate kinase of step 10 be turned up by F16BP, the substrate for the aldolase reaction of step 4? Well, let's remember that the difficulty of the aldolase reaction going forwards. The aldolase step, number 4, is always at risk of running backwards. It can be kept running forwards as long as substrate concentrations are high and product concentrations are low. If pyruvate kinase is not active, PEP accumulates and plugs the pipeline. When the plumbing stops working, things accumulate in your sink. This includes the products of reactions 4, 5, 6, 7, 8, and 9. Now that's great if you don't want glycolysis and the aldolase reaction to run, but if you do, you better call a plumber. That comes in the form of reaction 4's substrate, F16BP. When the aldolase of reaction 4 isn't running forwards, the reactant F16BP builds up. As its levels increase, F16BP binds to pyruvate kinase and activates it. And though it may be tempting to think of reaction products, such as those of reaction 4 being isolated from reaction 10, the fact is that the cytoplasm where the glycolysis occurs is literally a sink. Proximity is not a problem since enzymes, reactants, and products all mix together. Biochemists call it a soup, and it really is. Everything is mixed together. So when F6BP activates pyruvate kinase, as it can easily do, the drain gets unplugged, reducing the PEP concentration in step 9, which in turn reduces the concentration of all the products going back to aldolase's reaction in step 4. With the PEP clog removed, the reactions run in the forward direction as they need to here. Activation of pyruvate kinase by F16BP is called feed-forward activation because an intermediate in the pathway is signaling an enzyme further along to start working. F16BP is the Drano of glycolysis. Although we've been focusing on glucose, glycolysis is also important for metabolism of other sugars like fructose, mannose, and galactose. Fructose, for example, easily enters by adding an N-phosphate to become F6P, the same molecule we saw in reaction 2 of glycolysis. The enzyme that puts a phosphate to the end of fructose is hexokinase, which we already met at step 1, where it helped glucose to do the same thing. Hexokinase can also convert mannose to mannose 6-phosphate, and then another enzyme converts mannose 6-phosphate to fructose 6-phosphate. Galactose joins by converting through 4 steps into glucose 6-phosphate, and that's how it joins the pathway. So each of these sugars are readily converted into early intermediates of the glycolysis pathway, and then they can get made into pyruvate. Now, just as there is more than one way to enter the glycolysis pathway, there are also alternatives about what happens at the end. In particular, when there's no oxygen, there's an alternative exit from glycolysis. One such exit ramp is called lactic fermentation. It produces lactic acid, which is used to make foods like yogurt or sauerkraut. It's also the path used by our muscle cells during prolonged, vigorous exercise, when the oxygen supply to muscles is unable to keep up with the demand. Lactic acid was once thought to be the culprit in the muscle soreness that develops after heavy exercise, but studies have shown that soreness results primarily from microscopic damage to muscle fibers. A second fermentation pathway occurs in other yeasts and bacteria when they run out of oxygen and they produce alcohol from pyruvate. This is the exit ramp from glycolysis that wine and beer makers depend on. In this 2-step process, pyruvate is first converted to acetaldehyde and carbon dioxide is released in the process. The acetaldehyde is then turned into alcohol using electrons from NADH, thus regenerating NAD+. If you consume alcohol, your liver cells have the same enzyme as yeast, but it catalyzes the reaction in the reverse direction to make acetaldehyde from ethanol. For anybody who's ever had a hangover, this is a big deal because it's acetaldehyde that accumulates and leads to hangovers. Eventually, though, the acetaldehyde gets converted to acetyl-CoA, which gets oxidized in the citric acid cycle. So far, we've focused on what happens to sugar, but sugars are not the only source of energy in our bodies. In fact, they're not even the primary energy storage molecules. Those would be fats, also known as triglycerides or triacylglycerols. As you learned earlier, a triacylglycerol is a molecule that binds 3 fatty acids, or 3 acyl groups, to a glycerol. To get energy out of a fat, the acyls in it must all first be cleaved from the glycerol that they are attached to. This reaction is catalyzed by enzymes called lipases. These acyl fatty acids are a primary energy source for many tissues, including heart muscle. The fatty acid acyls enter cells with the help of membrane transport proteins. All entering acyl groups into a cell first get linked to a common cellular handle known as coenzyme A, creating a molecule we call acyl-CoA, or an acyl-CoA thioester. It is these acyl-CoA molecules that undergo the subsequent steps in fatty acid oxidation. Glycolysis was like an open highway in the cytoplasm, but fatty acid oxidation is smaller, more like a bike path. You might think of it also like a Pac-Man, chewing off 2 carbons at a time off of the acyl group as it operates. In eukaryotes, 2 cellular organelles are the controlled sites for fatty acid oxidation, the peroxisomes and the mitochondria. In plants, fatty acid oxidation is carried out entirely in the peroxisomes, which are organelles specialized for oxidation of substances. In animal cells, including our own, peroxisomes merely start the job if the fatty acid has 22 carbons or more. Fatty acid chains that are shorter go straight to the mitochondria. Peroxisomes in our cells cannot completely oxidize those long fatty acids. They merely trim the very long chain fatty acids to a more manageable size and then export the smaller pieces to be dealt with by the mitochondria. Most fatty acids of animal cells get oxidized in mitochondria. So let's follow what happens there. Upon arrival at the mitochondria, acyl-CoAs bearing fatty acid chains of up to about 20 carbons swap out their CoA for a molecule called carnitine to create acylcarnitine. Now this switching of partners is temporary. Acylcarnitine, but not acyl-CoA, has a transporter that will let it into the mitochondria. Once inside the mitochondria, the carnitine is swapped back out for a CoA to recreate acyl-CoA. And it is the acyl-CoA that enzymes involved in oxidizing fatty acids look for. The importance of carnitine in this big switcheroo, however, cannot be overstated. Deficiency of carnitine has health consequences, particularly for tissues like heart muscle that are heavily dependent on fatty acids for energy. Now, both in the peroxisomes and the mitochondria, fatty acid oxidation happens in cycles of 4 reactions, with each cycle yielding a fatty acid chain that is shortened by 2 carbons. This continues until ultimately we have only acetyl-CoAs. The oxidation of fatty acids is also known as beta oxidation because all reactions involve the fatty acid's beta carbon 2 carbons in from the carboxyl end. The first reaction involved in beta oxidation is known as acyl-CoA dehydrogenase, and it catalyzes the removal of 2 protons and 2 electrons from the bond between carbons 2 and 3 of the fatty acid. These are donated to the electron carrier known as FAD to create FADH2. There are 3 remaining reactions in fatty acid oxidation. Water is added first, with a hydroxyl getting added across carbon number 3. This removes the double bond that's there. The next reaction oxidizes the hydroxide that was just added from water and generates an NADH. And the last step involves the splitting off of a 2-carbon chunk known as acetyl-CoA from the fatty acid chain. The enzyme catalyzing this step is known as thiolase. The remaining acyl group from which the acetyl-CoA was removed gets attached to another coenzyme A, creating an acyl-CoA 2 carbons shorter than the original one. This becomes the substrate then for another round of oxidation. Each time the pathway is repeated, it chops off an additional acetyl-CoA so the fatty acid gets 2 carbons shorter with each round until the final carbon-CoA molecule is split into 2 acetyl-CoAs. Now at this point, we're done with fatty acid oxidation, at least for fatty acids that had an even number of carbons. Oxidation of fatty acids with an odd number of carbons, though, yields a 3-carbon CoA at the end. This 3-carbon CoA molecule is converted into a 4-carbon molecule known as succinyl-CoA, and it gets oxidized in the citric acid cycle, as we will see in an upcoming lecture. The enzyme that creates succinyl-CoA needs vitamin B12 as a coenzyme to help with catalysis. Based on this role in energy production, vitamin B12 is often added to energy drinks targeted at young people. There is no credible evidence, though, that the added vitamin B12 plays any role in increasing energy. Excess vitamin B12 is simply excreted. Now, most people get plenty of vitamin B12 from their diets, but the vitamin is not made in plants, which is why vegans who avoid all animal-based products need to eat fortified foods or take a supplement to get their vitamin B12. Now, speaking of diet, a popular fad is the ketogenic or keto diet. In the keto diet, 50% to 75% of calories come from fat, with the remainder coming from proteins and carbohydrates. The keto part of the name is short for what are known as ketone bodies. Now, ketone bodies are molecules produced by the liver when it uses fats as an energy source, when glucose and other energy sources are not available. The body would normally use glucose, but the low carbohydrate intake on this diet does not provide enough of it. Since we only store enough glucose and glycogen to provide a 24-hour supply, other backups must be in place. Ketone bodies from the liver are alternate fuels for the brain and other cells when glucose is not available. ketone bodies may actually alter brain chemistry, as it has been known since the 1920s that a strict ketogenic diet with virtually no carbs reduces the incidence of some types of epileptic seizure. So what do you think? Could an all-fat diet prevent synthesis of glucose altogether and help you drop the pounds? Well, not so fast. When the body runs out of dietary protein to make glucose, it starts digesting its own protein like muscle for the same purpose. That's the same thing that happens when people starve to death. You can stave off the muscle loss with a high-protein diet because many amino acids can be converted to glucose in a process called gluconeogenesis that we'll discuss in Lecture 15. But there's a price for that too. As we'll see in Lecture 19, ammonia is a toxic byproduct of such diets and must be handled by the kidneys. Fatty acid oxidation is ultimately about burning fat, a goal that never seems to go out of style. The acetyl-CoA produced by each cycle of fatty acid oxidation can directly enter the citric acid cycle, the same series of reactions that's fed by glycolysis. So both pathways lead there. The products of sugar glycolysis and the products of fatty acid oxidation both bring us to the start of the citric acid cycle. In beta oxidation, it just occurred to me. The process all takes place between carbons 2 and 3. Some hydrogens are first removed to FADH2, then water adds across the bond, the H to carbon 2. Hydroxyl oxidation's next, a ketone for carbon 3. Then thiolase, the enzyme, dissects the last 2 Cs. The products of the pathway, of course, are acetyl-CoAs. Unless there were odd carbons, then succinyl-CoA.